Hey everyone, welcome back to Windows Seat Studies. So, till now we have been learning about histone modification. In today's class, we are going to learn a very distinct and interesting type of modification, that is histone tail clipping. Till now, we have learned about histone acetylation, histone methylation, histone phosphorylation, as well as histone ubiquitination. All of those different types of histone modifications had one thing common in them, and that is addition of certain chemical group on the amino acid residues of the histone. But histone tail clipping is very interesting example because in this we are not adding anything. Instead, we are removing something. So we are literally cleaving the histone tail, and that is why it is called histone tail clipping. So this word has a literal meaning. So in this modification, we cleave N-terminal tail of histone. So the histone tails are cleaved in the histone tail clipping. So this modification is a very radical way of removing the histone other histone modifications, right? So all of the histone modification that we learned till now, they were thought to be permanent, they were thought to be irreversible. But then later at some point of time, researchers got to know that they are rather dynamic. But histone tail clipping, in which we are literally removing the N-terminal tail, this is an irreversible mark. So this is again a different thing from all of the other histone modification, that this is an irreversible mark, or we can say it is a permanent mark, because once you cleave the N-terminal tail, so the protein is changed forever. So for it can't be reversed. So either you have to make new histones. There's no other way from escaping from histone tail clipping. So this is that is why this is an irreversible mark. So histone tail clipping was first seen in tetrahymena. These are protozoans, the unicellular ciliates. So this was seen in 1980 for first time, and in tetrahymena, the first 6 amino acids of the H3 and terminal tail were removed. So this is now seen that is histone tail clipping is also seen in yeast as well as in mammals. Okay, so in yeast, the proteases that do the histone tail clipping, they are not yet known. But in mammals, when the mice embryonic stem cells were being studied by researchers, they found out that there's an enzyme known as cathepsin L, and this enzyme was seen to be performing the cleavage of N-terminal tails. So they found out that there's an enzyme known as cathepsin L that cleaves the N-terminal tail, and this cleavage of N-terminal tail results into cell differentiation. So this is how there was a discovery of the enzyme that performed the cleavage of N-terminal tail. But in the yeast, that is yet to be identified. The proteases that bring out this change. So with this, we are done with histone modifications. So let's do a quick recap of all of the histone modifications, so that we will remember and we will be able to summarize everything. So histone modifications are the modifications done on histone. Okay, those are post-translational modifications because obviously they are done after the translation. Post-translational means they are done after the synthesis of protein. So once histones are synthesized, the certain chemical groups are added on the histones, and that is what we call as histone modifications. So but now we learned that not only chemical group addition is identified as histone modification, but the cleavage of the tails is also known as histone modifications. So the mechanism of functioning of histone modification is of two main types. First, it can directly perturb the structure. Okay, means addition of certain group can directly hinder the structure, the chromatin structure or the nucleosomal structure, or it can act as a recruitment signal for certain factors which will change certain chromatin factors which will change the chromatin environment. So means addition of the that chemical group doesn't directly hinders the structure, but it forms it serves as a recruitment signal, or it serves as a docking site or the docking surface for certain chromatin factors, for certain chromatin remodeling factors, or some group of proteins which are going to bring out the chromatin environment, which are going to change the chromatin architecture. For example, polycomb group of proteins, so they bring out the silencing of gene, while the trithorax group of proteins, they maintain the active state of genes. So recruitment of such factors that can be done due to certain histone modifications. So these are two broad ways that by which the histone modifications can work. So the modification that directly perturb the structure are histone acetylation. The second one is histone phosphorylation. The other is histone ubiquitination. And the last but not the least, histone tail clipping that we learned today. Okay, so in histone acetylation, the acetyl group is added on the lysine residue, and the addition of acetyl group neutralizes the charge on the histones. So histones which are positively charged are now neutralized, and now histones are losing their positive charge. So this directly changes the interaction between histone and DNA, and this loosens up the chromatin and makes the DNA more accessible, and that is why histone acetylation is universal positive mark, oh sorry, universal active mark. So it leads to the activation of gene, and this is how acetylation directly perturbs the structure. Okay, so acetylation is done on lysine residue, and it is an active mark. Okay, and it neutralizes the positive charge of histone. The histone phosphorylation, this modification, in this modification we add phosphate group, which is overall negative group, and this is how this changes the histone DNA interaction. And this is done, the phosphorylation is done on serine, threonine as well as tyrosine residue with the help of different type of enzymes. The acetylation is done by the HATs, okay, and the deacetylation is done by HTACs. So addition of acetyl group is done by HATs, removal of acetyl group is done by histone deacetylases. The histone phosphorylation is done by kinases, and the removal of phosphate group, it is done by phosphatases. Okay, the histone ubiquitination, we add ubiquitin moiety. So ubiquitin is a 76 long amino acid long polypeptide. So this is a large covalent modification because we are adding a huge moiety on the histone, and now this directly changes the structure of chromatin. And this ubiquitination is done on 2' sites on H2A K119 as well as H2B K120. So this is an repressive mark, while this is an active mark. Okay, so those are antagonistic to each other. And the H2AK119 methylation is important for H1, the linker histone binding, while H2BK120 is an active mark. The other modification that changes the chromatin structure is histone tail clipping. So in histone tail clipping, we directly cut the N-terminal tails. So that is why we are changing the chromatin structure. So now those histones those histones are permanently changed. So this is an irreversible mark. The other mode of functioning is recruitment of factors, and this is followed by again different modifications. The first one is histone methylation. Okay, so we have discussed histone methylation in a very in great details. Okay, so we have distributed the histone methylation in six parts, in six different parts. We discussed in that about the enzymes which add the methylation or which remove the methylation. Okay, so the enzymes which add methylation are known as HKMTs if they are adding on the lysine group, but if it is done on the arginine residue, those are known as PRMTs. Okay, and the enzymes which does the demethylation, those are known as histone demethylases. They remove the methyl group. Again, the histone demethylases are uh, they only do the demethylation for the lysine residue, but in the arginine residue we have not found any methylase which sorry, any demethylase which can remove the arginine methylation. Rather, we refer the PAD4, which is a deaminase, because it replaces the methylated arginine with a citrulline, which is non-standard amino acid. So we can't directly say that it is demethylation of arginine, but we can still consider it as a demethylation because we are obviously removing the RMEs, the methylated arginine. But this methylated arginine, which is replaced by citrulline, we do not know that how the citrulline is then replaced by arginine. That is why we are not sure, or we do not call PAD4 as a demethylase. It is an deaminase. But there was an enzyme known as JMJD6. Okay, the investigation is still to be done, and this was found out to be doing the demethylation of arginine, but this is still yet to be investigated. So these are the different enzymes that does the histone methylation. Oh yeah, in the histone ubiquitination, the E1, E2, E3 ligases, they do the ubiquitination part. They add the ubiquitination. So the E3 ligases that does the job, that that ubiquitinated the K120 residue. Those are of two type that we learned. One was ring 1B, the other was 2A HUB. Okay, those are two different E3 ligases. There are enzymes that remove the ubiquitin group, and those are known as isopeptidases. So isopeptidases, they remove the ubiquitin group. So again, there is de-ubiquitination, and that is done by isopeptidases. Okay, so in histone methylation, those are the enzymes which are involved. Then again, in histone methylation, we learned about the specific methyl marks such as H3K9 methylation, H3K27 methylation, H3K4 methylation, H3K36 methylation, as well as H4K20 methylation. We learned about all of those in detail. So this is how does they recruit the factors? The H3K9 methylation, which is hallmark of heterochromatin, this recruits HP1. This provides a docking site for HP1 proteins, heterochromatin protein 1. The H3K27 methylation with PRC2 complex, this results in the gene silencing. So this is how all of those different methylation marks, they recruit certain factors which result into the chromatin architect change. Okay, but alongside histone methylation, there are other examples which can also recruit the chromatin factors, and those are histone acetylation as well as histone phosphorylation. Okay, so it is not necessary that a single that any histone modification should work through a single mechanism of functioning. For example, histone acetylation can directly perturb the structure, or it can also recruit certain factors. This is same in the phosphorylation case. It can directly perturb the structure, or it can also recruit certain factors which are going to change the chromatin. So this is the so histone acetylation and histone phosphorylation, how will they work? How will they change chromatin environment? This is all context dependent. For example, histone acetylation, it neutralizes the positive charge, but it also recruits factors, the bromodomain containing proteins. Okay, the proteins that has bromodomain, all of those they can bind to the acetylated histones. Okay, while chromodomain containing proteins, the proteins like HP1, they can be recruited on the methylated histones. Okay, so this is how this is how the histone acetylation and phosphorylation can work through both of the modes, both of the modes of functioning. So with this we conclude the histone modification part, and this was a, a summary of what we have learned in histone modifications. So with this we are done with histone modifications, and in the next lecture we will learn about long non-coding RNAs. So this is a very important topic. I'll cover it in detail. After that, we will also cover dosage compensation. We will discuss genomic imprinting. We will also discuss the polycomb group of protein as well as trithorax proteins. So with this, we will be and after that we will be done with the epigenetics series. So I hope you all are enjoying this series, and I'll see you in the next lecture. See you. Bye bye.